Thank you very much. Thank you, Jeremy. Thank you, Ruth. Thank you, Professor Yao. So, this is a small room. People at the back, if you want to sit down, I would suggest now is a good time to find a seat, unless you like standing. It's because it's a small room, I would invite you to interrupt, to ask questions, to do it like the physicists do it, especially if I say things that you think. I'm also going to give you a word of warning. The word of warning is because of the vagaries in the English educational system. I stopped studying mathematics 4 more at the age of 60. Okay? And it was no great tragedy either because I'm sort of competitive. I was top of my class in physics and chemistry and biology, and I was third in math. It turns out that the kids who were ahead of me were the children of professional mathematicians, whereas I was the child of a historian who paid me to add the grades of his students average. I wasn't being taught how to do improper fractions in school. Anyways, so I'm going to talk about looking for a place for theory in biology. And I'm here, as Jeremy mentioned, because I am, for reasons that are hard actually to explain, the director of the NSF Simon Center for the Mathematical and Statistical Analysis of Biology, and this is linked tightly to the Quantitative Biology Initiative in the Faculty of Arts and Sciences, which is run by Shar Ramanathan and Vinny Manoharan. The membership of the two things overlaps extensively, and the whole goal is to build an open and transparent community that tries to bring people from the mathematical sciences and biological sciences together to think beautiful thoughts and perhaps actually do some stuff together. Okay? No, why doesn't this work? Okay. So let's try it. Okay, so one of the points I want to make is that one way of thinking about biology is that it's a form of computation. So there's a definition from John Hopfield that computation is any rule-based system to transform one set of symbols into another. Here's an example. This is one of the first things my daughter was told to say. And I think you can think of most biological activities, if you define computation in this way, as forms of computation. And I'm going to talk, especially in the second half of this talk, about the idea that evolution selects for computations that increase fitness in current and future environments. I'm going to make a point about the relationship between the past and the present. Everything that you know comes from the past, right? Even as you're listening to me, my mouth moves, there are vibrations transmitted through the air, they act on your ear, your brain has to process your signals, so there's a time lag between me talking and you hearing stuff. When you hear stuff, you may decide this is all BS and want to raise your hand and ask some question from the MRC Handbook of Awkward Questions. There's a gap between those two things, right? And you have to predict things about the future. What if I say something as you've got your hand up that totally removes your objection, right? Now you have your hand up, and I go like, oh, so what have you got to say? Like, oh, the question disappeared, right? And so this is true for all sensation in biology. All information comes from the past, because sensory processes take time, and all action lies in the future. And the gap between the present and the future is a variable length for various different sorts of sensation. And so what evolution is selecting for is how to use information from the past to make accurate predictions about the future. Okay? I think there are important similarities to computer engineering and mathematics. So the syntax of computers is ones and zeros. The syntax of biology is A, G, C, and T. Okay? And both computer programs and biological programs have purposes, right? They are functions that they are designed to perform for organisms so that we can spread our genes in time and space. For computer titans so they can increase their fortune, money, and time. Okay? I also want to point out that it's not clear there's ever going to be magical induction. This is a picture. This comes from an old talk that I pulled the slide out of this morning. This is how people used, in the era that Jeremy first came to Harvard, to look at gene expression. These are little dots. An experiment has been done where two conditions are being compared. And if a dot is red, a gene is being expressed much more in one of the conditions than the other, and if it's green, the reverse is true. Okay? It is not clear there's ever going to be a way of looking at all this sort of stuff and coming to an inductive understanding of something like this. So this is just a diagram. These are chromosomes. They're attached to microtubules, which are railroad tracks that the chromosomes move along. This cell structure inside this is called the mitotic spindle. It's about to try and segregate those chromosomes. It's unhappy because this little dot here is not attached to one of these railroad tracks, and now you're not going to guarantee that these two sister chromosomes go in opposite direction. There's a checkpoint, a machine that can detect the difference between the red and the green dot, and amplify the signal. It can inhibit this little Pac-Man here that is going to degrade a protein that is going to induce the cells to divide. It's my contention that it's neither clear that it will ever be possible, nor that it will be worthwhile to imagine that just looking at this sort of data is going to tell us this sort of thing. Okay? So here's one sort of sense. The American version is called Rube Goldberg. The English one is called Keith Robinson. This is some very complicated scheme that somehow is going to get someone's cup of tea up on the top, right? And lots of people think about biology this way. It's this unbelievably complicated set of things. You have somewhere between 20 and 30,000 genes, depending on who's doing the counting. All sorts of craziness goes on. There are all sorts of backups of backups of backups. But there are at least principles that appear to make sense. So this again, this is a picture of a chromosome that wants to segregate itself. These are the centers that organize microtubules. The way you try and connect the trains to the tracks, it's as if you started in New York and you wanted to build a railroad line to Boston, and you just lay track for 200 miles. At the end of 200 miles, if you haven't found Boston, you just tear it up and start again. And you will eventually get to Boston that way. It's not the most efficient way of running a railroad, but it is a way of running a railroad. And that's what these microtubules do. So they grow and they shrink. People like Joe Howard work on this process. And they have the property that if they touch the chromosomes, they feel a bit like they got to Boston and they're less likely to shrink. And if they touch this special part of the chromosomes, which is where the locomotives are that will move the chromosomes along the railroad tracks, they are stabilized much more strongly. And you can imagine that even if these guys grow out in random directions, that preferential stabilization will eventually lead to this structure here. So the locomotives are attached to the tracks, and if you now break the linkage to hold the sisters together, they will dutifully move off in opposite directions, and you will have segregated your chromosomes. So this is the principle of exploration with selection, and it applies for many different things in biology. It applies to how proteins fold. It applies to how the spindle assembles, which is this diagram here. It applies to how your nervous system was formed in embryogenesis. You grew nerves in more or less random directions. Some unlucky ones did not, at least the ones that are designed to make your muscles contract, did not land on muscles and eventually withered and died. And other ones landed on muscles and received from those muscles survival factors that kept them alive, and that's how we can do this. Okay? It is hopefully, obviously, the principle of evolution by natural selection, and it probably has something to do with the behavior of ecosystems. So this is a useful principle that applies at many different scales of space and time. And one of the questions that I'm particularly interested in is whether there's a way of translating principles like that into mathematics. Right? It's easy to say for me, like, okay, I can argue this is a general principle. Is there a useful mathematical formalism that comes from that and encompasses somehow the idea that this is a scalable process? and a general process for doing things in biology, and there's obviously an interesting question for biologists, like how many other principles are there like this? Okay. We haven't found very many. Okay, so here's a different thing that I would argue less convincingly, like you should doubt me about this, that it's an organizing principle. It's things that are organized in modules. Just the way in Word, there's a spell checker. You could remove the spell checker from Microsoft Word and it would still work. You just have to proofread a lot more carefully. Right? It's a module. Right? There's a module inside your cells that drives an engine, a biochemical engine that makes the cell go through the cell cycle. It has a bunch of stuff. I'm not really going to explain it here. It's a biochemical oscillator. Okay? And it can run totally freely. Okay? It can induce things in one phase of the oscillator, like the assembly of the spindle. And in something like a fertilized frog egg, it does that and the spindle does not talk back to the oscillator. So the oscillator keeps running whether the spindle's assembled properly or not. In fact, the oscillator keeps running even if you take the DNA out of the cell. Okay? Work for Mark Kirschner and his colleagues. But in your cells, mine, and in our friend the humble budding yeast, there's another module, the one I showed you the picture of the thing you can't magically induce, that tells that these chromosomes aren't properly hooked up like these ones. It activates this machinery, and this machinery feeds back and inhibits one of the components of the oscillator, so the oscillation stops, and it will stay stopped until the chromosomes have lined up properly. Okay? All right, so here's an outline. I'm going to give you some thoughts on theory. I'm going to give you a brief introduction to this NSF Science Center for Mathematical Biology, just so that you may see the faces of people you wish to talk to. Okay, and you should, and we love talking to people, and we actually even listen occasionally. I'm better at talking than listening, but I have been trained by various people, including my wife and children, to listen at least some of the time. So I'm going to talk about the idea that maybe biology is not as underneath the hood is maybe not as complicated as we think, and then I'm going to tell you a piece of biology because it's cool and it's interesting and it relates to this question of evolutionary prediction. Okay? So here's an example of actual real theory in biology. This is the work of Michael Desai and Daniel Fisher. Michael was a graduate student with Daniel Fisher. He's now a faculty member in organismic and evolutionary biology. Daniel Fisher is at Stanford. And they wanted to ask how reproductive fitness, the mean reproductive fitness of a population, which population geneticists use the letter W for, changes over time. Okay? So they're very good, they're physicists, they go, well, I'm going to write down a function. So this is a function of all these parameters. Okay? And I guess the parameters may or may not be on the next slide. So I'm going to tell you what they are. NE is something called the effective population size. It's the sort of time averaged in a complicated way size of the population. UB is the rate at which beneficial mutations appear. UD is the rate at which deleterious mutations appear. Rho of S is the distribution of mutations of different effect sizes. S positive means the mutation is helping you. S negative, less than 0, means it's hurting you. There's some distribution. It's hard to measure. There are theoretical arguments about what it should look like, but in general, and especially when this work was done, we have no idea. Okay? So what they did, then epistasis is how mutations interact with each other. It's like if I have mutation 1 and I have mutation 2, is my fitness the addition of the values of the fitness contributions of both of those mutations? Is it just S1 plus S2, or is it some more complicated thing? Turns out, quite often, it's additive, but in important cases it's not. And we have, since we don't know what this is, we sure as hell don't know what this is. And then there are all the complications of sex and recombination. So, what Michael and Daniel did is, so this we call this the British assumption here, okay? And then they assume there's no deleterious mutations. The epistasis is of the form where you just add the effect of the fitness of the different mutations. the population size is constant, and every beneficial mutation contributes exactly the same amount. Okay? This is crazy, right? There's no way evolution works like this. Okay, and then, you know, pencils and papers get churned, some of the paper ends up in a trash bin, but eventually you get a result. It's complicated. Okay? I'll remind you, N is the population size, S is the contribution of each individual beneficial mutation, UB is the beneficial mutation rate. You get a formula like this, it tells you that the speed is proportional to S squared times the logarithm of the population size, and the speed increases, I won't say linearly, but sort of monotonically with the logarithm of beneficial mutation. Okay? And then these guys came and talked to me, and they're like, we should do an experiment. Yeah, sure, do an experiment, but like, we don't know how. So Michael crossed Oxford Street and did a very demanding experiment in my lab, and proved himself to be an outstanding experimentalist. And here's the result of that experiment, okay? I'm going to try and, I don't think this is a thing that you should take home and remember, but I'm obliged to explain the graph, so I will do my best. Okay, this is the average speed of adaptation. This is percentage increase in fitness per generation of the population. This is the relative value of the logarithm of the population size times the beneficial mutation rate. This is the prediction of what would happen if each mutation fixed individually and spread through the population before the next one did. That means that the rate of adaptation will go linearly in N times UB, which is why this curve looks crazy like this. Okay? Michael did the experiment with two different mutation rates. The wild type are in black. The mutated guys have a mutation rate which has been increased by a factor of about 10 are in red. His theory, the predictions are the open circles, or the open triangles, and the experimental results are the filled symbols. Okay? There's real variation amongst these experimental results because these are 6 parallel experiments. They're not actually going to go identically because that's not how evolution works. But what you can see is the fit with the theory, considering the wild assumptions that got made, is surprisingly good. And then there are interesting questions about why is it true that experiment fits theory so well when theory has made such crazy assumptions. And we have some understanding of why that is, and I don't think that understanding would have arrived had the theory not been generated and the experiment stopped. Okay? So maybe this is a good example of how theory can be useful in biology. Okay? But the real question is, can we do this all the time? So here's another example about progress. So here is J.Z. Young, who is an English zoologist. He worked at the biological station in Naples because it was warmer, it was better. And he worked on the squid and discovered this thing called the giant axons. So the axons are the wires, the nerve wires that your body uses. The giant axon in the squid is like 50 times bigger in diameter than the ones that we have, and it makes it a really useful experimental subject. And then these two English physiologists, Hodgkin and Huxley, studied, often in Woods Hole in Massachusetts, the properties of electrical conduction in these things. And this is one of the great triumphs of mathematics in biology. It's trotted out on a ridiculously regular basis. Many of you in the audience have seen it trotted out before. These are a set of equations. This is for the current through the membrane of these cells, and these differential equations describe the change in the number of open objects that will transmit either sodium or potassium ions. And this is some complicated cooperativity thing. And it turns out they could experimentally determine the values of these constants, show their spectacular agreement with experiments, and make predictions and verify the predictions. And the existence of N and M predicted the existence of molecular channels that admit either sodium or potassium ions through the membranes of cells and have complicated regulation by the voltage across the membrane. And this is Rod McKinnon, who won the Nobel Prize for discovering the structure of those channels. So again, this is a sort of example of as good as it gets. Okay? Here's some other examples. there are examples from circadian clocks, particularly bacterial circadian clocks, where they're very satisfactory mathematical models that correspond to biochemical reality. The work of Howard Berg in MCB over, I guess, in Caltech, at least 40 years, has provided an elegant description of how bacterial chemotaxis works that unifies both phenomenology, okay, so they swim about, and when the concentration of good things is rising, they swim in straight lines for longer, and when it's falling, they swim in straight lines for shorter periods. When they finish swimming in a straight line, they tumble wildly and start off again in a random direction. So what they do is find the good stuff by a biased random walk, because the length of the runs depends on whether the concentration of the good stuff is going up or down. And this is a truly spectacular achievement, and Howard is a truly spectacular person. Okay? So what do these things have in common? So I would argue the things that they have in common is that they are single and well-defined phenomena. So it's easy to define the phenomena and make phenomenological measurements. And you can at least start by fitting theory to phenomenology rather than worrying about molecules. And when you do it with phenomenology, you typically have smaller numbers of free parameters, and it's therefore easier to test whether you're being an idiot. Okay? People have figured out an appropriate level of abstraction at which to study the problem, and as I showed with the example of Michael Desai, simplifying assumptions have turned out to be enormously productive. And because of those simplifying assumptions, and because some of the people were prepared to roll their sleeves up and do experiments, or at least form productive collaborations between experimentalists and theorists, there was real linkage between experiment and theory. And the other thing that's interesting about these examples is they all use simple existing mathematics, right? So one of the things that NSF is not being particularly clear about with us and the other three centers is whether they want us to use existing mathematics to make sense of biology, or whether they want us to identify new mathematics, and you can actually get different people from NSF in the same room, in the same afternoon, saying diametrically opposite things from each other. I think there's an interesting possibility as an armchair observer and mathematical idiot. I'm not idiots, I'm just idiot. I think there's an interesting question about whether the difficulty of thinking about biology and this idea of how does one embody a principle in a mathematical formalism might call into existence new forms of mathematics. And I think it's at least worth having an open mind about that. Okay, so one of the questions is, what do the people in this room want? What do NSF want? What do scientists want? So one question is we want just more of these spectacular examples for single phenomena. We'll sort of count them up on our fingers as we solve individual problems. Maybe we want even more accurate models for the things we've already succeeded at. Maybe we want to change the level of abstraction for some of them, either to go down more molecular or to go up, to go from organisms to populations. Maybe we wish to be relevant to the real world. Here's the Purell if you're anxious about all the people whose hands you've shaken already this morning. My claim is that now my children are college age, I'm permanently immune to almost anything because they both on daycare at one point or another. And we live in challenging times. Despite opinions on Pennsylvania Avenue, the world does appear to be changing climatically. Okay? But maybe we want to go beyond single phenomena, like how do cells as a whole work? Right? Don't bother me with chemotaxis, around the spindle checkpoint, how do cells work as a whole? And so there are two possibilities. One is a purely predictive one. We will learn how to measure everything. We have people at Google. There's a lot of Yoo-hoo there, and that's the chocolate flavored drink that's popular with computer programmers in the past. And they will make purely predictive models. They won't necessarily satisfy our curiosity, but we will no longer just know what sort of shoes we want to buy when we log onto our computers, but we will go once a year and have 10,000 metabolites measured in our blood, and our pharmacist, not our doctor, will tell us what we need to do about it. Okay? And the other one is we'd like to satisfy curiosity, and I obviously wouldn't be here if I wasn't in the second camp. Okay? So here's the big thing we need to do, and this will serve as an introduction to the people in this NSF Science Center. We want to take genetic networks. This is a genetic network of genes involved controlling the fate of mammalian cells early in the development of mice and people. They end up making decisions about cell fate. So here is a stem cell, and it's going off to make different sorts of cells here, which are just cartoons, but they're different sorts of cells in your body that do different things. These make tissues, which make organ. This is the extracted gut of a mammal. Okay? And these are the morphologies of the guts of a whole series of different sorts of creatures. This is an alligator. The other ones are hard to read. This is a finch, right? And you can see there's all sorts of crazy differences, and those crazy differences have been selected by evolution. So we'd like to know at some level, what is the underlying program that makes this coiled structure, and then how do you tweak the dials on that program to make different sorts of guts and different sorts of animals, because they eat different sorts of things. So what we'd like to do is understand the emergence of functional phenotypes. We'd like to know about molecular circuitry. We'd like to know about the form of organisms. Someone will mention the fact that it's the 100th odd anniversary of someone who was worried about form and function, and whose book gets shown all the freaking time. It's not clear to me in retrospect. I read that book as an undergraduate. It was inspiring, but it's not clear he really made a fundamental contribution to,
he inspired a lot of people.
He inspired a lot of people, yes. Let's leave it at that. He inspired a lot of people. His name, I guarantee, will be mentioned by somebody. Okay? Then we worry about function, and we worry about all these things in multiple scales and multiple systems, so we want to go from gene to cell to organism to environmental adaptation, both within the lifetime of individual creatures. I heard people whining about the cold on the way in. You'll whine about it less as the winter goes on. Okay? And we want to do this virtuous cycle, assumptions, model, experiment, loop, especially using statistical methods. Okay? And one of the things I think is really true about biology is that the statistics that I was taught as an undergraduate and then forgotten, had to be taught again in graduate school, was designed for the world of R.A. Fisher when the number of measurements were small and the measurements were very noisy. We now, at least for a lot of biology, live in a different world where the number of measurements is almost infinite, right? And maybe we're more interested in how much of the behavior an individual variable explains, rather than the p-values of whether this set of 10,000 variables is statistically different from this other set of 10,000 variables, because they don't have to be different by very, very much to return p equals 10 to the minus 10. And so our goal is to have this layered understanding. There are all these arrows that connect things together, and it's cool and groovy. The interactions between these layers have all different forms. They're chemical, mechanical, electrical, optical. And again, ultimately, they're mathematical, right? That these things, it ought to be possible to embody all these connections in mathematics. And the question is how to do that. So here are the people in the center. It's led by 5 people. This is Cassandra Extevor. Everyone except me has at least 1 degree in mathematics. Okay? Cassandra is a developmental biologist, but her undergraduate degree from Toronto was both in mathematics and biology. This is Maha in applied math. This is Sharad Ramanathan, who's a refugee from theoretical physics. And this is Johan Poulsen, who was originally trained as a biologist, but thinks and is very qualified mathematically. So there are a bunch of people involved in math. There are a bunch of people involved in decision making. There's obviously overlap. These are people who work in morphogenesis. They range from Victoria D'Souza, who works on the structure of RNA molecules that control the fate of viruses, to Ethan Garner, who worries about the structure of whole bacterial cells and all sorts of levels in between. And then 3 of us are interested in this question of adaptation, and I mentioned Michael and I mentioned Cassandra. So we have science goals. We want to connect layers, find new principles. We want to make predictions. We want to exploit extant and hopefully develop new math. And especially what we want to do is produce collaboration between mathematicians, statisticians, and biologists. The money from NSF and science is being spent almost entirely on people. Okay? So we're trying to build a trainee-led community that would have an infrastructure for learning across disciplines. We are recruiting independent postdoctoral fellows. We have 3 at the moment. Ajaraza, Gautam Reddy, and the third person's name that comes to me, I'm really sorry. Okay? And we want to try and interact and influence through workshops and visitor programs. So in the back row, we have one of our visitors, Danny Forger, who's from Michigan, who's a mathematician, who's interested in circadian clocks. Okay? So, in the next 15 minutes, I'm going to try and tell very quickly 2 stories. Okay? So the first question is whether life might be simpler than we think. Okay? Right? You know, what's the minimum that we need to do to make a self-reproducing living thing? Okay? And my argument is the fact that organisms compete with each other requires them to get better and better at doing things, because otherwise they will get beaten by other people who are better and better doing things. It's just like the technological development that led the arms race between the Soviet Union and the United States and ended up putting people on the moon. And so here's what competition has done to our friend the telephone. Right? This is a direct lineal descendant of this. But, you know, high school physics students can understand how this works. And you can understand some general principle of how this works, but I guarantee there's no human being who understands how every single part of an iPhone works, right? Like there's some system engineers who are like, well, someone tells me how this system works and it interacts with this system, but there's no one who knows what every node in the printed circuits inside the phone. Okay? So I'm going to try and simplify 2 complex problems. One's maintaining cellular composition and one's responding to variable environments. So maintaining cellular composition, cells are complex systems. This is the density of parts inside a Boeing 787. This is the density of parts inside a yeast cell. The ratio of those 2 densities is about 6 times 10. And the proportions of the parts matter in both the yeast cell and the airplane, right? You know, the ratio of engines to airframe is sort of important for aircraft. Okay? So there's sophisticated circuits that control the synthesis and destruction of biological components. But those circuits themselves have to be regulated. And so there's this question of who regulates the regulators and then who regulates the regulators of the regulators, and you can easily worry about going off into some infinite hierarchy. Okay? So here's a way to think about this from first principles. There are 6,000 genes in budding yeast. Let's imagine, just for the sake of argument, there are 10 parameters per protein that's being expressed. That means the dividing cell will be following a path in 60,000 dimensional space, right? This one dimension per parameter. How similar are the paths of different cells? How unimodal are the distributions of the parameters for cells at any one point in the cell cycle? We have no idea. The short answer to that question. And we don't have any idea of how to measure it, because, you know, maybe at the same time we can measure the abundance and localization of 4 or 5 different molecules inside a single living cell. Okay? There's all sorts of fancy measurements we can do to compute the mean values of many, many things across populations of cells, but those methods are all destructive. The methods that will allow us to follow things in real time in individual cells are very limited. Okay? And there are all sorts of questions about how cells transit from very different states changing the value of many, many of those parameters all at once. And the question is, how much is this like taking your child bowling for the first time, where first time they swing the ball down the lane, you put buffers there so the ball doesn't go into the gutter, right? And the gutter equivalent for a cell is the parameters getting to some point where you're dead. And what is the machinery to keep cells from doing it? Okay? I think we have fundamentally no idea how that works. Okay? So now what I'm going to do is I'm going to do a bit of self-abasement. Okay? I'm going to tell you how poorly and stupidly I have thought about a problem and how a little bit of life came in how sometimes thinking hard but simply can be done. So I worked as a graduate student on the segregation of plasmids, small DNA molecules that are self-replicating that cells have. For example, I did it in yeast, but in bacteria they carry antibiotic resistance genes. Okay? And DNA is special. You can't make DNA except from pre-existing DNA. Okay? And what that means is you must never lose the last molecule of DNA. If you lose the last copy of a plasmid, if a cell is born without a plasmid, you're never, at least with that, by that cell just dividing and reproducing on its own, you're going to reintroduce that plasmid. You can't make a plasmid from nothing. And so people like me worried a lot about segregation mechanisms because you have to make sure you never lose that last molecule. And they're complicated things. High mean copy numbers are no protection because the copy number of every plasmid molecule just replicates once per cell cycle, will exhibit a random walk, and the distribution will spread out, and eventually some of the cells will have very few, and then they're liable to give rise to progeny that lack the plasmid. Okay? And there's all sorts of cool biology about how you don't just make sure that the mean copy number is high, but you restrict the variance of the copy number. Okay? And this is putting this in numbers. If one cell's copy number is 20 plasmids, the probability that you generate a daughter, a particular daughter, doesn't have any copies of the plasmid is 2 to the minus 20, which is 1 in a million. But if the population mean is 20, the probability of loss is much, much, much greater than that. It's actually a somewhat complicated mathematical problem, so as a graduate student, I taught myself how to do computers. Okay? But proteins aren't like DNA. You make RNA from DNA and then you make protein, and therefore it's okay to lose the last copy of a protein because you can make new copies. Okay? And here's the change in the number of molecules of X. There's some synthesis rate, there's some destruction rate times the concentration of X. And deterministically, at steady state, the concentration of X is just the ratio of these two rate constants. But molecules are indivisible, and they're made one by one, and so even though this is deterministically true, the concentration is going to fluctuate because destruction and synthesis are stochastic, and so temporally the number of molecules of X will be a one-dimensional random. Okay? And you can worry, and I used to worry about cells making different molecules at different rates. Right? So this is like having different rates of compound interest. Okay? If the cell isn't growing, these lines just keep diverging. Okay? And I used to talk to graduate students when I gave seminar and say what a big problem this was, and everyone has to have this appropriately scaled synthesis rates. And then much more recently, I came to my senses and realized that when cells grow and divide, they are effectively destroying molecules. That is, they're diluting them because the volume of the cell increases. And what that means is if here's some molecule where we're trying to make 100 copies per cell, and we have the synthesis rate of this one, and we double the synthesis rate of that one, and this is just deterministic, you change the ratio of them, but you reach a new steady state ratio. Okay? So yes, you have to worry about the ratio, but the concentration of these proteins don't diverge indefinitely over time. You just reach a new ratio. And so people are worried about keeping error at bay. So this is Howard Berg, who with Ed Purcell did a really beautiful piece of work figuring out the balance between how long you sense conditions for and how accurately you sense them. We did very elegant physics on this. This is my colleague Johan Paulsen, and Johan constructed his favorite diagram here and used this to make a complicated but compelling mathematical argument about, given the number of the things being regulated and their regulators, the best you can do whatever Maxwell-ish demon you have there to do the regulation. Okay? And I know about both of these things, and I'm sat on the thesis committees of Johan's students who worry about whether things get segregated binomially or not. But I had partitions in my mind, so I was thinking about things completely differently. But when I finally came to my senses, I was like, I can't do what Howard or Johan can do, but I can do a computer simulation. So I'm going to show you a slide of computer simulation where cells aim to make 100 copies of 100 different proteins. Okay? They make the proteins stochastically in each cell cycle. They segregate them binomially when the cells divide, and they just keep doing it. There is no regulation whatsoever. Okay? So it's 100 different protein species. You make 100. If you're making poissonially, you make 100 plus or minus 10 per cell division. And here the protein abundance is over 201 cell division cycles. Okay? And what this shows is in each cell division cycle, the copy number of the most abundant protein, the copy number of the median abundance protein, which satisfyingly is around 100, and the least abundant. And this line is the 91st most abundant, and this is the 10th most abundant. Right? And what you can see is without any fancy regulation at all, these numbers are surprisingly well constrained. And therefore, if you imagine different activities being determined by the ratio of different proteins, those activities are not going to change very much, right? And so one assertion that you could make is, I don't need any regulators. And so I think, at least for molecules that are made in reasonable quantities, I don't think this is why we bother to regulate. I think we bother to regulate to match the cellular composition to the environment. Because if you're a yeast cell or a bacteria, the environment that you're in can be very different from the environment your direct ancestors were in, and you might want different ratios of proteins. Okay? So the last thing I'm going to talk about, I'm going to talk for 3 more minutes, so I'm not going to get to the second thing, is what the cells know about their environment. Okay? You have more senses than you think. You can list 5, but your body knows what your blood pressure is right now. Okay? And if you stand up with your hand on your pulse, you will notice that your heart beats faster for about 5 seconds. And that's because your blood pressure went down. Your body knew that the blood pressure went down because when you stand up, the blood has to travel further from your legs to get back to your heart. And your heart is adjusted to beat faster, all without you knowing a single thing about it. Okay? Now you're worried about what will happen. Okay? So microorganisms have a particular form of sensation, and I'm going to show you a picture here. Their form of sensation, they have these molecules called histidine kinases that respond to various things in the environment. They take a phosphate group from ATP, they put it on this molecule called a response regulator, and that typically, but not universally, regulates gene expression. This is how bacterial chemotaxis works, for example. Okay? Each pair of these systems is a unique sensory module. Your typical bacterium has about 40 of these, but there are bacteria that have as many as 200. So that means 200 different senses, right? So we can ask the question about how you integrate global responses. So imagine you have 40 of these 2-component systems. Imagine that the cell cares about 3 levels of activity: no activity, medium, and high. There are 3 to the 40th, which is 10 to the 90th different combinations of activity. That's one big-ass lookup table, right? And that lookup table, that's, it's many more bases than many more combinations than there are bases in the genome. So there has to be some form of integration or arbitration. This is how many characters there are in the books in white for comparison. Okay? So cells have to do some form of dimension reduction, and at least as far as I know, we don't know what that is. Right? So I will leave you with two options. One is that there's a central processing unit. You have a nervous system that's doing all this computation. We have neuroscientists, we have systems neuroscientists, and they will understand consciousness and all sorts of other stuff. Right? So one of the possibilities is that microbes have a protein-based central processing unit that does all this integration and arbitration and does computation instead of using the lookup table. You could ask, how could it work? How many components would it need? What would the phenotype of the mutants in this thing be? And how could we have missed it? I think we could have missed this. Yes?
How do you define components?
Different sorts of proteins. How many individual genes that make proteins? Thank you. Exactly. Right? But the second option is that there's a single processing rule. The whole system is much dumber. So like I said, every pathway has 3 output levels. The lowest output level says, things here are cool, do not change anything. The second says, let us make a local homeostatic response. Let's say we detect reduced pH outside. We induce activities that make us better at throwing protons out of the cell to try and keep the pH inside the cell from going down. And if we can do a pretty good job, maybe the pH goes down a little bit, but it doesn't go down so far that anything bad happens. And the third level of response is code red, right? System pulls the fire alarm, cells stop growing, you induce all these emergency responses, and you just try and wait it out. Right? So if that was true, then all you have to do is combine the signals from these various pathways, because at this level of action, you don't have to do anything. Here, you're just making local changes. Right? You know, it's like the car. You can turn the heat up and down, and nothing changes, right? Or things in the rest of the car change a little bit, right? It changes the probability that the fan comes onto the radiator if you're running the heat. Okay? And then various things happen. If the wheel falls off, you're supposed to stop. So here are different ways of combining those signals. One is that you respond at the level above this only to the strongest signal. You can have 12 different pathways, each making local corrections, but as long as none of them have hit code red, the cell doesn't hit code red either. You could respond to the sum of the signals. So each of those 12 pathways would have to be active quantitatively to a 12th the level that any one of them would induce code red, but now you just add up their signals, and it's like 12 different systems are concerned, there must be something bad, let us stop. And there can be some sort of nonlinear response. And so the last thing I'm going to leave you with is that this is accessible to experimental tests. Right? The experiments actually ask what the rule here is are remarkably simple, and I keep threatening to go to my colleague Mike Loud's lab. Thank you.
So we have a little time for questions, I think. Anyone want to open the batting? Come on, guys, we're here for 3 days. We might as well get started.
Yes.
Could you give a summary of the second thing that you didn't talk about?
Yeah, I can give a very quick summary. What I was going to talk about, if I had been more disciplined or spoke faster, is the idea of bet hedging and the fact that, so if you take yeast cells and you grow them in abundant glucose, they grow happily. If you suddenly remove the glucose, you reveal heterogeneity in the population. So half the cells completely freak out, and they stay freaked out for a long time. And the other half of the cells, I shouldn't say half, a fraction of the cells freak out, and a fraction of them freak out much less. Okay? The ones that freak out were growing faster before you took away the glucose, and the ones that didn't freak out were growing slower. And what we think is going on is that there are two strategies. I'm going to grow as fast as I can. So I will throw all my reserves away to grow faster. Like if every time you divide, you have to store glycogen, so it doesn't matter if anyone feeds you tomorrow, you will have to eat more. You'll have to do more stuff each day, right? Whereas if you just eat exactly what you need, but you'll have to get fed tomorrow, otherwise you'll go into some nasty coma, you can grow faster, okay? And what Laura Baghammer, the person who did this experiment, showed is there appear to be these two different modes in yeast, and we can measure the rate at which you switch between, and they're heritable for some limited number of cell divisions among cells that are genetically identical. Laura can measure the rate at which you switch between them. Okay? And what we would, and if you take natural populations, at least they have different steady state ratios of these two phenotypes. So this is an example of what would be called non-genetic individuality or epigenetics. And she and Lee are, and we can make mutations that lock them in one state or the other. But what we are desperately trying to do, and failing so far, is to understand what the machinery is that stochastically puts the cells in one of those states or another, and we haven't gotten it. But that's what I was going to talk about.
I have
Yes.
a question. Yeah, so with regard to the simulation experiments you made with the cell division,
Yeah.
showing that the different level of proteins are maintained over cellular division. So is that a general, so that can be generally applied to all kinds of cell division? Because we know, for example, that the very amount of protein is partitioned in a very specific way to allow cells to adopt different fates, for example, in the case of stem cells, or even in yeast, in metabolic transitions, things like that.
Absolutely. Okay, so you're absolutely right. So I guess the argument I was trying to make is that things might, for simple cells which are just trying to make approximately identical copies of each other, things might be much more simple than we think. So I'm really interested in this question of, can we accurately figure out what the minimal set of things that a cells needs to stay alive? So here's the analogy I used. If you came from Mars and you wanted to understand automotive engineering, the best thing to look at probably would be a drag racer, okay? Because it has an engine, it has some big ass wheels, it has a transmission. If you take my car, right? We don't have the fancier model, it's a minivan. But it could have those flip down video screens to pacify the children in the back. And I can't start the car unless my foot is on the gas and the car's in park, because otherwise I might accidentally run someone over. It makes it, if you were just trying to understand a car from first principles, it would be way harder. And my guess is the fraction of young men and young women who grow up to work on their own cars has gone down enormously because cars are so complicated. And yeah, I used to be able to tune my Volvo, right? And so I'm interested, I want to know, I guess, I want to know what a Model T cell looks like. And can you do things in a more complicated way? Yes. And many of those things matter for exactly the things you're talking about, and they're cool and groovy. But I want to know what the simplest possible living cell could be made to look like. That's my big thing.
Thank you.
You're welcome. Yes.
So I have a question. You mentioned at the beginning that the history somehow, the information that, so I'm imagining it's somehow the cells are using that, let's say, to create these mechanisms to respond to environment, right? Something about the history or of the environment is somehow stored in the mechanism.
So,
And then what happens if the environment that you've seen in the past, well, the environment that you see in the future does not correspond to the things that you've seen in the past?
okay, so that's a great question, right? So, one way cells store history about their environment, long-term history, is by building sensation circuits and things like a circadian clock. Like the circadian clock was built by evolution. It makes predictions about when the sun will come, right? The question about, and in some sense the circadian clock is storing information about time history because it can be entrained. One of the questions I'm really interested in, I think we know very little about, is how many other things are there where cells physiologically store some part of time series history about the environment, right? Like this obviously goes on in nervous systems, right? But for unicellular things like yeast and bacteria, do they do some form of storage and computation so they have knowledge of what the glucose concentration was for the last 2 hours, right? If we were less lame about getting around to the experiments we promised NIH we would do, I promised I would do that experiment and we have yet to find a person in the lab to do it. But I think it's a super interesting question. And the simple argument you can make, so there's a beautiful paper from Ido Cassell and Stan Weidler, where they asked about whether cells should regulate the rate at which they switch between strategies based on information from their environment. And they can compute, like, if that machinery costs less than this, you should switch. I am sure that you can do exactly the same computation for the merits of storing time history about environmental variables and whether they help you prepare for the future. I don't think anyone's done it, but it has to be similar in spirit. And if the cost is less than something, you ought to evolve it, and we should check. But we're late; we don't get around to doing the experiments. But maybe I will go back freshly inspired because you asked the question. Yes.
In the end, you showed these two options, was central processing unit and a signal processing unit with nonlinear integration of the signals. What's the big difference between the two options if you have nonlinear integration?
Well, that's a good question. You could imagine a way in which these things. Let me say it this way, okay? Because you expose a appalling contradiction. The cell cycle oscillator for eukaryotes is in a sense a little bit like a central processing system. My colleague David Morgan at UCSF first made this argument, in the sense that various different inputs to it can stop the cell cycle in different places. Right? And I guess I meant by central processing unit something much more complicated than that. But I should make the distinction between what I'm proposing much clearer. Right? And I guess what I meant is that you can have things, different pathways interact with each other, modify each other's components, but every component is part of a pathway. So those interactions can make something that's akin to a central processing unit, but there are no components there just to act as part of the central processing unit. Whereas you can also imagine a system where there's a separate set of components that individual sensory pathways feed into. But its whole role is to just process, aggregate, arbitrate between those signals. But yeah, I should make the distinction clearer, sorry. Yes?
Because on that note, isn't that what you're describing, P53, the cancer protein that is supposed to tell the cell if it should give up and die or if it should prepare for everything's clear?
Yeah, I mean, I guess the point I wanted, yes, there are all sorts of things that cells have to process information, to integrate and arbitrate amongst some smaller number of things. I guess the point I was trying to make is at least in bacteria there's a very simple way of counting the modalities. So in eukaryotes, everything's sort of connected to everything else, which is its own interesting question. Whereas in prokaryotes, it's really true, it's this one-to-one connection, and the response regulators tend to have particular dedicated jobs, and therefore it's easy to set up this straw man. I mean, it's not completely a straw man, where the number of combinations of inputs from those things is ridiculously large, and therefore to propose, that at some level there needs to be some way of thinking about how you deal with the combination of those signals.
Just
may I add another question? So just extrapolation on this point. So by saying that there is a simple modularity to deal with that, do you mean there is something indeed mechanistically simple within the cell that do the job, or there is some structure within the cell that simplify things?
I don't know which of those things it's true. I mean, what the proposal, I mean, the specific proposal I made. So, okay, so it's good to go out on a limb and make explicit statements that will irritate people and then inspire them to prove you wrong. Okay? So I'm going to make the following statement about how I think this works in bacteria. The individual sensory systems or modules have 3 levels of output. You don't need to do anything. Make local corrections, and this is code red. And my prediction is the way code red is invoked is that any one signal has to be above the code red threshold, and if you have 2 signals that are halfway there, you do not go into code red, except possibly for some special combinations. And like I said, that's an eminently experimentally testable prediction. It's it's actually a surprisingly simple experiment. We just don't know the answer. But I'm gonna make that prediction. And you looking intently at me might actually get me to go over to MIT and do the experiment. Okay? Thank you very much.